Bom dia, boa tarde, boa noite aos internautas que nos acompanham neste programa Nanoalerta, hoje no seu número 180. Fazemos este programa dedicado ao público não especialista, porque é este público que paga as pesquisas sobre nanotecnologia realizadas no Brasil. Também fazemos este programa porque é aqui que nós podemos discutir os impactos da nanotecnologia na sociedade, no meio ambiente, na economia, na ética, no direito, coisa que não são discutidas aonde se faz nanotecnologia no Brasil. Por fim, fazemos este programa porque acreditamos que ser livre é ser bem informado e a divulgação científica contribui, colabora para o aprofundamento da democracia e da cidadania no Brasil. Hoje, conforme tínhamos anunciado, vamos nos conectar agora com o professor Dr. Andrew Smith. Vou chamá-lo no Skype, que ele já está nos esperando lá. Hello.
Hello, this is Andrew.
Yes, very well. I have
Hello,
your
Paulo.
sound and your image very well. Okay?
Excellent.
I would like you introduce yourself. Tell for us your experience with nanotechnology.
Certainly. I'm Andrew Smith. I'm a professor at the University of Illinois at URBNA, and I've been working in nanotechnology since about 2002. So that's when I started graduate school. And
Yes.
graduate school, I did work in nanotechnology.
Yes, a very long experience.
Yes, definitely.
Okay. So,
Hello, are you still there?
yeah.
Okay, great.
You will use PowerPoint or not?
Sure. I have slides.
Yes, you can share the slides.
Oh, I see. Okay, let me try to do that.
Yeah.
Share slides.
I think there is a tool that is share.
Yes. Okay, I got it. Did that work?
No. Not yet.
Okay. Let me try again.
But there are another possibility.
It says I'm sharing my screen. Did that? Okay.
You have to share for the Skype. The other possibility is you send me this PowerPoint by email.
I sent it to you about 2 minutes ago.
Yeah. 2 minutes ago. I will see.
Okay. Let's see. Share.
Yes, it's here.
Great. You got it?
Yes, I can put it in the screen here.
Excellent.
And you make your comments, and at the end of your comments on the slide, you tell me the slide number 2.
Okay,
the slide
that sounds
number
good.
3, and I put in the screen. Okay, I will open here.
That sounds great.
Yes, I have to put it there. Where is? Quantos dots nanoparticle for biomedical imaging?
Yes,
Is this?
that's right.
I have to see how to put it. There. Okay, first is the screen.
We're on the first slide.
Yeah, is the title.
Great. So the focus of what I work on is a quantum dot. So that's that first word, those first 2 words, quantum dots.
Yeah.
They're a specific type of nanoparticle that has been widely used and developed since about 1981. And so they're expected to, at some point in the next several years, win a Nobel Prize based on this invention.
Nobel Prize is Dr. Sturded.
Right. There's nobody's won a Nobel Prize specifically in quantum dots yet.
No. Okay. So you can continue. Is the next second?
The second slide is a picture of a quantum dot. It's a nanocrystal that's about 10 nanometers in size. So they're super tiny, but they're also, as you can see in the image, they look like a crystal, meaning that they have a regularly arranged pattern of atoms. So that's an important aspect of them. They're crystalline semiconductor materials. So they're like silicon, made out of that's what's in a computer processor, of course. So they're like this, except they're independent and can be dispersed in water and dispersed in solutions, so they can be useful for biology.
Okay. Very interesting, this image.
So that's an electron micrograph. The third slide.
Yes.
That shows this really key feature that has made them so useful.
Yeah, it's different lengths of the same material.
Exactly. Different lengths of the same material have different colors, and they emit light, so you can see them.
What is the material?
This is cadmium selenide, CdSe.
Okay.
So you can see that they're dissolved in liquids in this example, and they have different sizes, and so we can make them in different colors, which is a unique feature. Traditionally, to make something fluorescent in a different color, you don't really have an ability to tune it. You have to develop a whole new material, but this is something that the material is always the same. All you have to do is change the size, which is really easy to do, actually.
Yeah. It's the size matter.
Exactly. And so this is called the quantum confinement effect. This was discovered in 1981.
Okay. The next is 3 pictures.
So this depicts some of the advanced technologies that use this. So in the last 3 years or so, quantum dot televisions have been sold on the market by Samsung and QD Vision. And so quantum dots are the light emitting components of these screens. They're also part of the Amazon Kindle Fire. So these screens and display devices that emit light, that you can see, of course, now the light emitting components of those are quantum dots. And the big feature of those is that they have really crisp color. They have really sharp colors that you couldn't get otherwise. Also, quantum dots are also used in LEDs, and in future solar cells, most likely, they're being integrated into solar cells as well. So these are some of the everyday types of tools and technologies that these have been made into in terms of a pretty successful commercial venture.
Yeah, here in Brazil, in TV, there is an ads of Samsung that white contours dot is not good. Our television there is not with white contour dots, only another colors.
Oh, really?
Yeah.
Yeah, the ones they have here, it's not blue. You only have green, really good quantum dots that emit in that wavelength range. So they're not every single 3 colors in it. So it's not the blue, green, and red. It's really just the green that's made a big difference so far.
Okay. The next.
So this slide shows all these other types of applications of these materials that are not based on at-home right?
Yes, is slide 5.
Yes, exactly.
Okay.
So the major goal of using these in biology for biomedical imaging is to locate the tissues and image them and analyze them. So these have made a really remarkable improvement in our ability to analyze and image tissue and cells that come out of biology. So this is what we focus on in our work, is the use of these for imaging tissue and imaging cells. We can get a lot of information out of this that we could not get otherwise. We could not see the level of detail and read out the number of molecules that we need to see without this technology. It's been pretty revolutionary for biology as well.
And what is the nanomaterial?
Say it again, please.
What is the nanomaterials that you are using in this case?
It's the same thing. It's cadmium selenide,
it's the same.
CDSE,
okay.
Quantum dots.
Yeah. You think that is the best? Cadmium is the best?
Well, so far, nothing has replaced it. It's been the best since 1993, and it's really because it allows you to have a lot of different colors, and all of the colors are really sharp, meaning that they're narrow bandwidth. They're very distinct colors. Nothing else has given us that ability yet. They're theoretically not the best. Other ones should be better, for one reason, that cadmium is toxin. And most of what we do, it doesn't matter if it's a toxin, because we're not using it in any scenario in which there'd be toxicity. But ideally, it would be made out of materials like silicon or carbon or things that are more biocompatible. But those materials are not good yet. They don't emit narrow band light yet. They're not. The one key feature of these that's been so great is that they are extremely stable and they emit very distinct colors. And nothing else has been better than this yet. But in the future, I think something else will be better.
Okay, my question.
Sorry, I lost you for a second. I keep breaking up.
Yes, my question was about the nanotoxicology, about cadmium, but I think you answered. Huh?
Right. This is extremely important. nobody really understands exactly whether these will be toxic in the body. But it is known that when you put cadmium-based quantum dots in a mouse and you wait, they don't leave the body for 2 years. They're still in the body, in the liver and in the spleen after 2 years, so they don't get excreted. But there are ways to get around that. It's believed that if you make them really, really small, they will be filtered out of the kidney and they will leave into bladder. Also, if you make them biodegradable out of non-toxic materials, they probably won't cause problems. But nobody's solved this problem yet. This is a really, really interesting field, and it's there's a lot of basic studies that need to be done before these are used in human beings.
What is the very, very small in this case that you told?
6 nanometers.
6 nanometer.
Right. There's a very distinct filter size in the body. If things are smaller than 6 nanometers, they can move throughout the whole tissue in the body. So right now, most nanoparticles like this are in the range of about 10 to 30 nanometers, and so they only have access to blood. If they're below 6 nanometers, they can move through the blood vessel surface, which is the endothelium. It has a pore size around 6 nanometers. So things that are smaller than 6 nanometers can pass across this and easily get out of the kidneys into the bladder and into the urine. So that's the goal, is to make these really small, smaller than 6 nanometers, but it's really hard to do.
And why we doesn't have basic science in this area that produce more knowledge about the toxicology problems?
That's a good question. There are a lot of people working on this, and they do really good science. It's just that even beyond this, the whole field of biomaterials toxicity is a lot of challenges in it. A lot of it's because you do work in a mouse, and it's not a human being, so we don't know exactly if it translates to being used in human being the same way. The biology is different in a mouse versus a human. It's a rodent, of course, and we're, of course, not rodents. Also, it's a challenge to study this in detail because of the difference in how we study toxicology in a lab versus actually what's happening in the body. We normally study things when you take cells out of the body and you do cell culture, and you basically have isolated individual cells, and you can get a lot of great studies out of that. But it doesn't tell you what's happening in the liver and the spleen and the lymph nodes where these materials end up. It's really difficult to get really good data out of that, to really analyze it in detail. There's details missing in that type of study. So I think eventually we'll be able to monitor these more effectively and see what actually is happening in the body. But currently, the limitation in the tools of the toxicology tools is a really big problem.
And do you think that?
Sorry, you broke up again. Can you repeat that?
And do you think that with the computer simulation we can solve these problems or not?
Yeah, I think so. Computer simulations certainly help in many aspects. There's a couple levels of that. You can simulate the material itself. This is a lot of what we do with the collaborator in material science in our university. We simulate materials so we can figure out which ones will be effectively replacing cadmium selenide. So trying to find out which one's going to be the next one that's not going to be toxic. Simulating toxicology is very, very challenging. Simulating biology in general is not as accurate as it is when you do it in crystalline physical materials, like solid state materials. So it's a big challenge to accurately simulate something in biology. But if people develop enough do enough studies, you can do data mining on that to evaluate what might happen with a nanomaterial. So simulation definitely helps, yes.
Okay. You can continue in slide 6.
So slide 6 kind of shows a depiction of what we do in our lab. About half of our lab studies individual cells. This is a single cell, and it's kind of comparing a single cell with what we look at like a textbook of a life science textbook or
what is the length of this single cell?
Yes, that's right.
What is the length of this single cell?
It's about 20 micrometers, so smaller than what your eye can see, but also it's about at least 1,000 times bigger than a quantum dot.
Yeah.
So a lot of what we are missing in biology is how molecules function dynamically. We know a lot about DNA and protein and where it's located, and we know a lot about analyzing its sequence and structure, but the dynamics, how they move and how they regulate things, is a big unknown. This is a lot of what we study in our lab, analyzing how cells receive a signal and how they translate that signal into deciding whether to divide or to move or migrate. These are kind of functions of how a cell works, and also how it changes when it becomes cancerous. And so we can analyze the signaling processes that are kind of invisible right now. That's a big function of how quantum dots are being used to map these processes and looking at the dynamics of them.
Okay.
So that's kind of like a really microscopic way these are used. On slide 7, the next one.
Yeah.
This shows how we use this to analyze human patients. These are biopsy tissue from tumors.
Yes.
And these allow you to basically get a profile of a disease. In cancer, you have genetic mutations. And these genetic mutations cause excessive cell growth and excessive cell motion, so that they move into surrounding tissues. So when somebody has cancer or some sort of mass, one of the first things that's done.
Yes.
And so we could use a biopsy together with quantum dots to give you a readout of the mutations that are there. That's what's depicted here. This is a prostate tumor. And different regions you can see that look very, very different when they're analyzed with quantum dots. And we can quantitatively analyze what mutations are there in different areas. So that's called.
Yes. Slide number 8.
So slide number 8 shows that traditionally quantum dots have been depicted on the top left. They're spheres. They're like circles if you look at the cross-section.
Yes.
But there's a huge number of different materials available, and we have not been using those. Almost everybody uses a sphere made out of cadmium selenide. But there's huge advantages to using these other ones. Rods, tetrapods, discs. They have really unique properties, and they can do very unique things. Like rods preferentially absorb and emit light out of their ends. And they'll also have what's called polarized light. You can potentially determine their orientation. Are they pointing towards you or away from you? Or are they pointing to the side? You can't do that with a quantum dot. So you have more information available to you if you use these other materials. But they're made out of different crystalline structures. If you go to the next slide, slide 9.
This is crystals, no?
Exactly, they're all crystals.
Yes. You make these crystals top down or bottom up process?
Bottom up.
Bottom up process.
Right.
Yes.
Yes, we do it in a chemical synthesis in a liquid.
Uh-huh.
You can also make them top down, so you can have them etched onto a surface of silicon. But then if you do that, it's really hard to remove them and to use them afterwards. They they would be deposited on the surface in that case. We actually put them into liquids and then put them into cells or into a tissue. So bottom up for us is absolutely the best way to go.
And it is very difficult to use.
Can you say that again, please?
It's difficult to control this process of bottom up.
It is, yeah. You have to really, really be meticulous in the synthesis, because it's not like synthesizing a chemical where you can synthesize it at 60% yield and then you can purify it. These are really hard to purify away from other types of contaminants. You have to control the kinetics really, really precisely, meaning that you have to begin a reaction where you have a tiny nanoparticle, you have to let it grow until it gets to a very specific size, and then you have to really, really rapidly stop the reaction. And that control actually has been one of the key limitations about how to really make these commercializable, because quality control is very challenging. This is one of the reasons that these have not really scaled in their production for biological use, is because of quality control issues. It's really hard to make these under many circumstances.
And about the geometry,
Say again, please?
About the geometry of these crystals.
Yes.
You have control. If you want to make a triangle, you can do this. If a circle, you can do this.
Exactly. You have very unique types of control. You don't get this out of other types of nanocrystals. It's really hard to control the shape and the size very precisely. One of the reasons you can is because these are ionic, meaning that you have atoms that are charged inside of the crystal, so that some of the atoms are positively charged and some are negatively charged. And you can use that to make these different shapes and sizes. If you try to do this with a material that's not ionic, that's purely covalent or metallic, it's really hard to change the shape really precisely. This is a very distinct capability of quantum dot-like materials, these ionic materials. You have a lot of shape control. And so you can study what happens in biology and what happens in different types of applications using these specifically. We know a lot about shape-dependent biological behavior because of these materials.
Okay, the 9.
So we can make different shapes, we can make different sizes. One of the reasons we can do this is because quantum dots, like cadmium selenide, as you can see on the left, are made of crystals.
Yes.
A pattern of atoms that is perfectly aligned. They actually come in two different, what's called a crystal phase, and that's the way in which the atoms are aligned. If you look at the two depictions on the right, these are showing the arrangement of atoms. Blue and red indicate selenium or cadmium atoms. And if you look at the two different patterns, they look pretty similar, but in reality they're slightly different. And these can exist in these two different forms. And because of that, you have a whole bunch of different shapes and types of properties you can get out by tuning the difference between these two. You can just pick from the beginning whether you want zinc blende or quartzite. So these two materials, you can select them based on how you do the synthesis, and they give you different properties at the end. So we've been publishing papers on this, how exactly you can pick out these different crystal phases and what the properties are. If you go to the next slide.
Please, and
Sure.
you use the zinc blind for what? And the wurzit for what?
There's almost no difference between the two in terms of their use. However, they give you different types of opportunities. Currently, the ones that are used in biology and the ones that are used for televisions, they're actually a mix of the two. Usually these two will actually form in the same crystal, and people have not really spent a lot of time trying to pick these out precisely. But the difference is, the really important thing, is that zinc blende, it usually makes a sphere. It usually makes a flat type surface, and so usually rounded to some degree. Wurtzite gives you the ability to make elongated structures like a rod and a tube. So zinc blende is more spherical when you make the crystal, and wurtzite is more like a rod, elongated.
Okay. The 11.
So
No,
one of the main
10.
Okay, 10. One of the main reasons it's hard to really precisely control this is the way that you analyze this is with a technique called X-ray diffraction. In X-ray diffraction, you typically have to have a large amount of your material, and you basically shine X-rays on it, and you see a pattern of how these are reflected by the crystal, and you get something that looks like these, what's called a spectrum, where the red lines and the green lines show you what this looks like for zinc blende and wurtzite. So it's hard to really see from this picture, but they look very similar. It's hard to distinguish these two. They are really challenging when you have a tiny nanocrystal to really determine exactly what your crystal phase is. And these experiments take like a whole day to do a few. It's really, really laborious and challenging. So on the next slide, what we just discovered is that you don't have to go through this X-ray process. You can do this with optics. Optics are really easy. You can take a measurement in about 20 seconds, 30 seconds. And also it's much, much more sensitive, meaning that you only need a really, really tiny amount of material in order to take a really robust measurement. And this is on the order of 10,000 times less material compared to what you need in X-ray diffraction type analysis. And so for quality control purposes and for use in making these manufacturable and useful, this is a pretty big leap forward for us, because you can actually have this in your process analysis. So you can just take a small amount, analyze it, and continue your reaction. You can actually get a measurement out in just seconds. So it's a pretty big advance for the use of these and for the analysis of zinc blende and wurtzite.
And you...
Say it again, please.
I ask if you are...
You broke up again, sorry.
My question...
Sorry, I'm still having problems. Can't hear anything now.
Yes,
Sorry.
I
Okay.
will write it. One moment.
Okay, I can hear you now, though.
Yes.
I see, yes. Yes, we're the first people to discover this. So it might seem like it's obvious to some people who work in a field like nanotechnology, where you're always analyzing the optical properties of a material. You're analyzing whether it absorbs light, emits light, and what colors of light it absorbs and emits. But this actually was a unique discovery, because the signatures of this, the exact reasons these emit light and absorb light distinctly based on the two crystal phases, is actually in the ultraviolet. People don't typically look at the properties that much in depth in the ultraviolet. The spectrum everybody cares about is the visible spectrum. The visible spectrum, of course, gives you the ability to make a useful material for televisions, for lights. This is what the eye can see, so everybody cares about this. So we actually studied these materials really closely in the ultraviolet, which of course is not useful for making a television, but it tells you tons of information about how the bonds are aligned. So all of these bonds in the crystal, the way in which they interact with the atoms, you can easily determine more information about the crystal nature by looking in the ultraviolet, because it's higher energy, and these bonds are distinct at higher energy. Sorry, I can't hear you. Maybe write again?
Yes, I'll write again.
Oh, I can hear you now.
Yes, if you have the patent of this methodology.
Yes. We have a patent application. It typically takes in the range of a year. So we published this in, I believe, April or May of this year. And so, yes, we've applied for patent for this. Of course, it's very hard to get patent in a methodology more so than a material. Of course, since it's really hard to enforce it. But yes, we have applied for an application for a patent.
Okay, the last, I think the last, no, the last slides.
Oh, the last slide is just the reference for this paper. It was published in Nature Communications several months ago. And this is a collaboration. This work we're very much an experimental group. We do work in synthesis, optical analysis of nanocrystals, and our collaborator, Schleifer, is the author, the second author of this. He's a computational material scientist. So everything he does is based on simulations. And this has been a really great opportunity to work with him, because he can tell us more about what we observe by simulating these crystals. And it's turned out that simulations are very accurate for these materials when they're done with a really robust technique called density functional theory. And he's an expert in this, and we get to basically recreate everything we observe, and we can tweak it in a mock simulation instead of having to create something from scratch. So this is a great approach to nanomaterials design, I believe, having the ability to do experiments, make something, analyze it, and then simulate it from basic physics, so from first principles of forces and quantum mechanics.
Okay. Questions for hello?
Hello. I can't hear you again.
Here, I put right.
Okay. Yes, questions? The budget for the research.
Yeah.
Yes. Most of what we do comes from funding from the National Institutes of Health in the United States. National Institutes of Health are the primary funders of research related to medicine and biology in the United States. And so everything that we do is driven toward those goals of making things practically useful for human health. The National Institutes of Health fund our work. We also are funded by the National Science Foundation. National Science Foundation in America primarily funds basic research. So things that are just for the study of fundamental science, for the advancement of science. So both of those organizations.
Okay.
How much money?
Yes.
These grants are in the range of about $1 to $2 million US dollars typically. And it really depends on the duration of the work and how expensive it is. In America, most of the money from a grant that goes to an academic lab like ours is an academic lab, so we're based out of a university. Most of the money goes to tuition for students and salaries for researchers. So the actual cost of doing research like this in terms of the materials is relatively cheap. It's pretty easy to synthesize something, and computational analysis is usually free. You pay for supercomputer time, but it's really low cost compared to personnel. So typically these grants are in the range of $1 to $2 million, and they last somewhere between 3 to 5 years. And that money primarily goes to paying for a researcher to work on this, like a postdoctoral fellow or a faculty, and also for graduate students to work on this in the process of obtaining a PhD.
Okay, and how much?
Say it again, please.
How?
Yeah, I didn't get that. Could you type it perhaps?
Yes.
So I guess this project has been ongoing for about 2 years, and we're continuing to work on this. So we're going to be working on this project for at least another 2 to 3 years. This is just the beginning of optical crystal structure analysis. So we've only looked at a couple materials: cadmium selenide, cadmium sulfide, and cadmium telluride. We're looking at a very broad range of these, and we're also looking for additional signatures in the spectrum, in the optical properties. We think that there's a lot more there than what we've discovered already. So we've been doing this for 2 years. Several more years in the future we'll be doing this as well.
Yeah.
The research team has been led by 2 labs. One is my lab, and one is the Andre Schleifer lab. And within these labs, my lab has about 12 people in it, but only a couple people are working specifically in this field of the optical crystal analysis. So there's 2 people in my lab that work on it. There's 1 person in Andre Schleifer's lab that works on it. So that's in total 5 people. But there's also a researcher in Korea that was a postdoc in my lab named Seung-Jun Lim. He's the first author on the paper, and he works at an institute called Digist, and he's continuing this work there as well. So the 3 of us have been primarily leading this work, and several other lab members. So 3 primary leaders, and then also about 3 other lab members too. folks type again? I can't, I'm picking up very little. Yes, thank you very much. I greatly appreciate your time, and it was a pleasure speaking with you.
Okay.
Great, thank you. I look forward to hearing from you again, and have a nice day.
You have also.
Thank you, I appreciate it.
I appreciate it.
Bye-bye.
Bye-bye. Thank you.